and how that could affect barrier function. When I heard he was moving into autism research, I was really excited, because this is someone who is, like I say, a world-renowned scientist and very well established. So it helped to have some of his caliber in doing the work with autism. So that was fantastic. And on a different note, he is a fantastic singer. Not that we will get a chance to hear that, but I have had the privilege of hearing him. All right, thank you, Dr. Fasano.
Thank you, Paul. No singing today. Thank you so much. Very kind introduction. Let me start to make a quick remark. I've witnessed in this past day and a half phenomenal science. And this was only made possible thanks to the support of the Brain Foundation and the donors that generously been, donating to allow all of us to really create this syndicate of, high risk, high reward, line of science that otherwise would not have seen the light of the prime time that we heard during this day and a half. So thank you again, Brain Foundation. Thank you, the donors, past, present, and futures, that will allow us to do this kind of science.
Thank you.
Thank you, Pramila, to make us, recipient on this, projects. Today, we will talk about a theme that I heard, recurrency, this triangulation between the gut, mucosal biology and immunology, and therefore antigen trafficking, the microbiome and immune system that affects this communication, bidirectional communication between the gut to the brain. And who's going to deliver our project is a very promising colleague of mine that I believe has all the rights to, eventually make the presentation of our project, that Dr. Marcy Kingsbury. She's an assistant professor of pediatric Harvard Medical School. She's also an assistant investigator at Lurie Center for Autism at Mass General Hospital, and also one of our most brilliant principal investigators in mucosal immunology and biology research center at Massachusetts General Hospital. Her line of research has been focused on the hormone oxytocin at birth and development nervous GI system, and she's also very much interested in genetic environmental risk factors of autism. In fact, how this will affect offspring in terms of gut and brain development. And lately, thanks to this project, the Brain Foundation, she's moving toward this gut-brain axis and the interaction between the microbiome and neurodevelopment. I have to say, this is complementary to a much bigger project that we have. This called GEMMA. There is a birth cohort of infants at risk of autism that will eventually fall over time, and with the multi-omic analysis and many of the stuff that you heard today. And I have sadly to report that we have recruited 100 kids, and 3 of them already have developed autism. So the incidence in this cohort is 1 in 33. This is very scary. So we hope that our science and science, the others, will try to slow down, if not stop this trend. So with that, Marcy, the floor is yours.
Thank you very much, Dr. Fasano. Can everyone see my screen?
Yes.
Okay. I also want to thank the organizers for the opportunity to speak on behalf of Dr. Fasano and our work today. I'm going to be talking about our two-pronged approach using a humanized mouse model and human gut organoids to exploit and interrogate the zonulin pathway for personalized treatment of autism spectrum disorder. I don't have any disclosures related to this work, but I do again want to acknowledge the Brain Foundation and the O'Sullivan Foundation for their generous support of the experiments that I'll propose and present to you today. So as we all well know, ASD has core symptoms that include impaired social behavior, impaired language and social communication, restrictive repetitive behavior, as well as increased anxiety and sensory deficits and sensitivities. I also want to mention that there are comorbid diagnoses with other disorders that are seen in individuals with ASD that are relevant to some of the research I'll present today. so that individuals with ASD, approximately 30% of them are also diagnosed with social anxiety disorder and attention deficit hyperactivity disorder. And as we've all heard in this symposium over the weekend, autism is increasingly recognized and has been recognized more and more as a whole body disorder with comorbid symptoms. And the ones that I'll be focusing on today are GI symptoms, which often lead to or associated with dietary and feeding issues, as well as immune dysfunctions, and has already been nicely iterated. These GI symptoms can include constipation, diarrhea, gastric reflux, abdominal pain, and visceral hypersensitivity. And they are associated with these symptoms in ASD individuals of irritability, social withdrawals, stereopathy, hyperactivity, and language regression. And as you've also heard, the severity of the GI symptoms can, in some individuals, correlate positively with the severity of the core behavioral deficits. So that led us to, again, further address whether there's a role for gut dysfunction in the gut microbiota in ASD. And I think in listening to the talks, this is not necessarily a question anymore, but GI symptoms and altered microbiota are observed in anywhere from 25 to 70% of children with ASD. In a small open label clinical trial, microbial transfer therapy was shown to improve both the GI symptoms and the ASD core behavioral symptoms in children with ASD with severe GI dysfunction when looked at 8 weeks post-treatment, and remarkably, the benefits persisted at the 2-year follow-up in these children. In addition, targeted microbial therapy has been shown to improve specific social behavior deficits in a variety of mouse models of ASD, and shown here in this paper by Segreti et al., they looked at both genetic, environmental, and idiopathic mouse models of ASD. So, what is the mechanism by which gut microbes or dysbiosis may contribute to core behavioral symptoms of ASD? And we would like to hypothesize that a leaky intestinal barrier allows the passage of pathogenic microbes, antigens, and food particles into the bloodstream, triggering inflammatory processes, immune activation or dysfunction, and altered social behavior. So when we look, we can find studies that have reported that individuals with ASD are characterized by increased intestinal permeability. And one mechanism that we're drilling down on for the increased intestinal permeability is the presence of the haptoglobin 2 allele, or the HP2 allele. And this is an allele that's linked to zonulin production, as zonulin has been identified as the precursor protein of HP2, or the proprotein. And while human haptoglobin exists as 2 common alleles, HPA1 and HP2, generating 3 genotypes, the presence of the zonulin gene is more often encountered in chronic inflammatory diseases. And when we look at individuals with autism, this haptoglobin genotype analysis revealed a trend for overrepresentation of the HP2 allele in individuals with ASD compared to healthy controls, and an overrepresentation of the HP2 genotype and an underrepresentation of the HP11 genotype in ASD patients, specifically with GI symptoms. So zonulin is the only human protein to date that has been shown to regulate intestinal permeability by modifying epithelial tight junctions. And zonulin causes tight junction disassembly through proteinase activating receptor 2 transactivation of epidermal growth factor receptor. And this cytoskeletal rearrangement and subsequent disassembly of the tight junctions is caused by zonulin occludins 1 and occludin moving away from the tight junction, creating leaky barriers. Thus, an HP22 genotype may create the greatest vulnerability for leaky barriers due to enhanced zonulin production. And zonulin has been shown to contribute to the pathogenesis of several inflammatory diseases associated with tight junction dysfunction. Shown here in this table are chronic inflammatory diseases associated with zonulin. I highlight in here the blue that are common inflammatory diseases of the bowel, In red are common neuropsychiatric and neurodevelopmental disorders associated with some form of zonulin involvement, and autism is highlighted here. Additional evidence to support the role of zonulin in ASD comes from measurements of serum levels of zonulin, which are elevated in ASD patients and correlate positively with ASD severity as well as GI symptoms in 2 recent studies. So in the first study, there was almost a 200% increase in serum zonulin in the patient ASD group compared to the control group. And in this 2021 study, autism severity as measured by a CAR score, or the Child Autism Rating Scale, correlated positively with serum zonulin levels. Now, it's known that both gluten and dysbiotic intestinal microbiota can trigger zonulin release in the gut, increasing the transcellular trafficking of bacterial metabolites and dietary antigens. And once released, zonulin can also be transferred to the brain, where it can increase blood-brain barrier permeability and trigger neuroinflammation. And neuroinflammation and increased blood-brain barrier permeability have also been described in ASD patients. So a team member, Dr. Maria Fortino, looked at postmortem tissue from individuals with ASD and found alterations in blood-brain barrier tight junction proteins and neuroinflammatory markers. And I'll show some of that data here. So she did find that when she looked at some of the tight junction proteins, such as claudin-3, claudin-5, and claudin-12, and tricellulin, she found that compared to healthy controls, postmortem tissue from individuals with ASD showed an increase in these tight junction proteins. In addition, she found increased expression of blood-brain barrier proteins associated with neuroinflammation. And two of these proteins were MMP9. MMP9 is a matrix metalloproteinase that cleaves extracellular matrix components and is believed to be involved in the pathophysiology of neurodevelopmental disorders. And studies have shown that this protein, the secretion of this protein can cause blood-brain barrier disruption. TSBO is a translocator protein located to the outer mitochondrial membrane and has been a target for neurodevelopmental and neuropsychiatric disorders. So as I mentioned, in addition to these blood-brain barrier tight junction protein alterations and alteration neuroinflammatory markers, we know that the bacteria and microbes within the gut of ASD patients can be altered. But it is currently unknown whether the gut microbiota from ASD patients with GI symptoms exacerbates the intestinal and blood-brain barrier permeability to contribute to core behavioral symptoms. So our hypotheses for the experiments I'll describe to you today that are being supported by the Brain Foundation is that a genetic predisposition for increased gut permeability conferred by an HB22 genotype and consequently an ability to produce zonulin synergizes with dysbiotic gut microbiota from ASD patients to induce leaky gut and blood-brain barriers, producing neuroinflammation and behavioral deficits. So I'd like to introduce you to our zonulin transgenic mouse and how we're using this mouse to demonstrate zonulin-mediated increased antigen trafficking that can lead to chronic inflammation. So wild-type mice only express the HP1 allele. So to examine zonulin-dependent increased permeability, we acquired a mouse model in which the native mouse HP1 allele was substituted with a murine HP2 allele via targeted insertion to generate mice of the HP22 genotype. When we look at these mice at their small intestine and their colon, they have preserved histology, but they have increased gut permeability as assessed by a fixie-dextran assay, where mice gavage the dye, and then we look at how much of the dye is in the blood, and that's indicative of how much of the dye is transferred through the intestinal barrier. And as you can see at baseline in both females and males, the transgenic HP2 mice show increased fixie-dextran serum levels as compared to the wild-type controls. In addition, when we look at their gut microbiota, through PCA analysis, we can see that ZTM mice, their gut microbiota, both the males and females, is clustering separately and differently from that of wild type. And when we look at the relative abundance of certain species, we find that ZTM mice show gut dysbiosis with increased pro-inflammatory Rickenella and decrease in Akkermansiae, which is a protective health-promoting gut microbe. We can also push the phenotype by administering dextran sodium sulfate to induce colitis in the drinking water of these mice. So when these mice are given DSS in their drinking water for 7 days, and then that treatment is stopped, a few days later, we can see 70% mortality within our HP2 zonulin transgenic mice given DSS compared to all the other mouse conditions. And following this increased mortality, we see DSS-induced permeability in ZTM mice. This is at baseline, not under conditions of DSS treatment, but the permeability is significantly exaggerated in our zonulin transgenic mice when they're given DSS. We can look at the histology of the small intestine, which looks relatively normal, but we can see DSS-induced colonic damage and inflammation in both our wild type and ZTM mice, but it's much more pronounced in ZTM mice. And it was interesting because in the wild type mice, one group showed persistent colitis with occasional mucosal erosion and crypt distortion, whereas another group of our wild type mice showed complete recovery after DSS treatment. But in the DSS treated mice, there was a homogeneous and severe inflammatory process with severe mucosal erosion and a loss of the regenerative crypt department. Quite remarkably, when we administer zonulin inhibitor AT1001, we can prevent the mortality and we can ameliorate colitis and intestinal permeability following this DSS treatment. So shown here is the original mortality that's induced with DSS. And when mice are administered DSS, but also the zonulin inhibitor, we can rescue that mortality. The blue line is now up here at 100%. We can also rescue the colitis through the administration of this inhibitor with the DSS treatment. And we can rescue the increases in permeability. So this again is the fixed dextran levels in the blood under DSS conditions in our zonulin transgenic mice and in the DSS conditions with the inhibitor. I'd now like to move on and just talk to you about what we see in terms of behavior in our ZTM mice at baseline. And we've just recently published a paper in Press and Tissue Barriers showing the behavioral phenotype of these mice. So when we look at restrictive repetitive behavior as assessed with the marble burying test, the number of marbles that they bury that are placed on top of their wood chip bedding, we see a significant increase in marbles buried in female ZTM transgenic mice compared to wild type, suggesting an increase in repetitive behavior. And when we place the mice in the elevated zero maze and measure the time in the open arms versus the closed arms, we see a significant decrease in our male ZTM mice for percent time spent in the open arms, suggesting increased anxiety. We've also placed our mice into an open field arena to look at general locomotion as well as measures of ability to spend time in the center versus around the sides. And while we see an increase in the time in the center in males, when we look at the traces for males and females, we think this time in the center is just due to an increase in locomotor activity. And it seems that the traces are much more enhanced and pronounced in both males and females than that shown by non-significant increases in distance travel. So it seems that these mice are hyperactive, and these are all under baseline conditions. Alba, within the lab, together with Maria, went on to look at the expression of blood-brain barrier tight junctions, and they found that in both male and female ZTM mice, compared to control mice, you find an alteration in the expression of tight junction proteins in males and females, occludin, VE cadherin, as well as in females, claudin 1, 3, and 5. And then in males specifically, they found an increase in 2 pro-inflammatory markers, IL-1 beta and CD11b. And I wanted to just mention that they focused in on claudin 5 protein expression in ZTM mice. And claudin 5 is a tight junction protein that's endothelial specific at the blood-brain barrier and is the most abundant tight junction protein. And here they found a significant increase specifically in ZTM transgenic male mice. And when Maria looked in the postmortem ASD tissue, she also found a similar increase in claudin 5 as compared to healthy controls within the cortex. So to summarize, before I move on to the experiments that we're directly working on, the ZTM mice are characterized by increased intestinal permeability and altered expression of gut and blood-brain barrier tight junctions, and increased expression of some neuroinflammatory markers and increased anxiety-like and repetitive behavior. And to summarize some of the human ASD data that others, as well as we, have discovered, individuals with ASD can be characterized by increased intestinal permeability, elevated levels of serum zonulin, altered expression of gut and blood-brain barrier tight junctions, and increased expression of some neuroinflammatory markers, and gut dysbiosis, as well as GI symptoms. So again, the hypothesis that we're testing with these experiments is that a genetic predisposition for increased gut permeability, the HP22 genotype, synergizes with aberrant or dysbiotic gut microbiota from individuals with ASD to exacerbate a leaky gut and blood-brain barrier, promoting neuroinflammation and behavioral deficits. So to address this hypothesis, these are some of the experiments that are underway with generous support from the Brain Foundation and the O'Sullivan Foundation. The first aim is to determine if stool derived from ASD patients exacerbates the gut and the brain inflammation, gut and blood-brain barrier permeability, and behavioral deficits in our zonulin expressing transgenic mice as compared to wild type mice. So this is a schematic of our behavioral setup. We have plans to mate female and male mice in January to generate the offspring that will serve for this experiment. We may enact an early life stress, maternal separation, to induce or to kind of push the phenotype similar to the DSS exposure. We will then perform antibiotic treatment to deplete the gut microbes in our zonulin transgenic and wild type mice, followed by fecal microbial transfer of stool collected from children with ASD with GI dysfunction and neurotypically developing controls. And so what this transfer looks like is that mice offspring of each genotype, HP11 and HP22, will receive exposure to both neurotypical pooled stool and ASD pooled stool. And as I said, we've been actively collecting stool and we're still in the process of doing that. Following the transfer, we will perform permeability assays, behavioral assays, and tissue assays of the brain, the gut, the stools, and the blood. The second aim that we're undertaking is to determine if stool derived from ASD children with GI dysfunction exacerbates small intestinal permeability, transepithelial antigen trafficking, and gut inflammatory processes in human intestinal tissue from ASD patients genetically predisposed to increased intestinal permeability, as opposed to ASD patients without this vulnerability. And so what this entails is that through a collaboration with MGH doctors for patients that are coming in for clinically indicated endoscopies, we will obtain duodenal biopsies, we will genotype the tissue, we will create intestinal organoids, we will create then intestinal monolayers where we'll perform permeability assays and inflammatory and tight junction assays. And we will do this under baseline conditions and then also following exposure to neurotypical stool as well as ASD stool. We currently have about 56 duodenal biopsies collected from both ASD and females and males, as well as a number of control males and females. And by late next week, we'll have the genotyping completed for these biopsies and be able to start creating our intestinal organoids. Perhaps the most exciting aim is to determine if the predicted exacerbation of barrier permeability and behavioral deficits that we expect to observe in ZTM mice after exposure to ASD stool can be rescued by blocking zonulin signaling using the zonulin inhibitor AT1001. So again, through the use of this inhibitor, we hope to prevent the opening and leakiness of both the gut and the blood-brain barriers. And so we will do this in the same behavioral paradigm, and the zonulin inhibitor will be added just prior to the fecal microbial transplant, and we will perform the similar assays. We will also do the same type of experiment using the inhibitor within our intestinal tissue. So here, to determine if the predicted exacerbation of barrier permeability observed in human intestinal tissue after exposure to ASD stool can be rescued by blocking zonulin signaling in our cultures using the inhibitor. And we expect the greatest exacerbation in our HP22 genotype intestinal tissue incubated or exposed to ASD pooled stool. And we expect the greatest amelioration of inflammatory impermeability by administering the inhibitor in that case. I just wanted to mention that it's exciting in that the zonulin inhibitor AT1001, also known as larazotide acetate, is currently in use in clinical trials. It's in a phase 3 clinical trial for celiac disease in adults, and it's also in a phase 2 clinical trial for COVID-19 multisystem inflammatory syndrome in children, known as MIS-C. And MIS-C is an inflammatory disorder that stimulates a massive immune response following SARS-CoV infection. And these children have elevated levels of SARS-CoV in their stool, in their serum, as well as elevated serum zonulin, suggesting a breakdown in the mucosal barrier integrity due to increased zonulin release that may facilitate the hyperactive immune response. So it'd be really interesting to know if this inhibitor can preserve that mucosal barrier and prevent perhaps that hyperactive immune response. The proposed impact of our experiments, these are what we hope we will achieve, is that to define the role of zonulin in gut blood-brain barrier permeability in mediating the effects of the microbiota on the development and progression of core ASD symptoms. We'd like to validate zonulin and high throughput haptoglobin genotyping as biomarkers of gut-brain permeability, allowing for the stratification of the ASD population to specifically identify subgroups of ASD children that may benefit from interventions aimed at correcting gut permeability. We'd like to validate the zonulin inhibitor as an interventional tool to mechanistically link brain function and behavior to synergy between zonulin and the gut microbiota in ASD children. And finally, we'd like to provide a basis for a future clinical trial to test the effectiveness of the zonulin inhibitor in treating and ameliorating core symptoms of ASD in affected children that can be stratified for zonulin expression and/or the HP1 versus HP2 genotype. So lastly, but not least, I'd like to thank the leader of our team, Dr. Alessio Fasano, as well as past members that have been instrumental in generating the data in our ZTM mice, Dr. Craig Sturgeon, Maria Florentino, and Alba Miranda Ribera. I'd like to just shout out to our current team assembled to conduct the experiments that I proposed today. And again, to acknowledge the generous support of the Brain Foundation and the O'Sullivan Foundation for making this work possible. Thank you very much.
Thank you very much. That was excellent. That was really very interesting work. So I think again we've got maybe we're running out of time. So one of what you could do is please email respond to the chats again and question and answers. Pramila, what is your view on?
Yeah, thank you so much
Is
for
this-
chairing this session, Paul. So we'll take a break till 1:30 because we are running late, but sorry about that. So we'll resume at 1:30. And I wanted to mention, we keep putting up these slides with information about if anybody wants to contribute to the Brain Foundation or support this research, we have a set of greeting cards that we can send to you. So please look at that. Thank you.
Great, thank you.